Viral interference in the ocean. A presentation by Professor Gershom Zajcek, Faculty of Medicine, Hebrew University of Jerusalem. We ask, what is the elementary unit of biology? Hitherto, we regarded cell as elementary unit of biology because this is what Rudolf Virchow taught. Cells beget cells, omnis cellula is cellula, and therefore cell is the elementary unit. But it's not so simple because you don't see any single cell in the environment. New cells never remain alone. They always join a family. And cell family is the elementary unit of biology. So how does such a cell family exist or be created? There is a clone founder which by dividing several times creates a family, and what's important in this family that they have the cells have the same self, and I shall discuss it immediately. And this is what is important in the family. So cell in a family carry a unique self in their genome, and virus is a messenger of the family self. Several years ago, Harry Rubin published a very interesting, exciting paper about viral interference. And I summarized this paper in another video, which is here, mentioned here. And here is a summary of the story. Rubin studied two viruses, RSV, Rau-sarcoma virus, and ALV, avian leukosis virus. And he studied their interaction, or what's called viral interference. What he found here, if you take an embryo of ALV, this embryo will resist the infection of RSV. So this is the essence of viral interference. And he called it RIF, resistance inducing factor, but this is the factor which induces viral interference, and RIF resist RSV infection. I described it in my previous presentation. So first of all, in order to compare it with what happened in the ocean, let's define what we are talking about. So what is a provirus? Provirus is a set of cell genes with three functions. It stores self of a cell, it controls who may infect the cell, and it controls viral production. Among other, this is a definition of a self in this family. Self is a set of provirus genes unique to a cell. So this is provirus, and so let's see what's a virus. Virus is a cell messenger. Virus is created by provirus, and virus stores self of the cell in the genome. Virus infects only cells with the same self. Again, self is a set of provirus genes unique to the cell, and here you have the first video which deals with this definition, theory of virus and cell update. So now we can study or understand viral interference. So we are talking about class 6 of Baltimore classification, where this everything happened, and ALV attaches to a receptor, and triggers the formation of provirus class 6. And this provirus controls whatever happens in this cell. And among other, provirus creates virions with the same self. But the essence of this study is that if you take RSV and this RSV attaches to a receptor, but it will never create virions. So this is a viral interference in a nutshell. And I think that this viral interference is very important not only for the clinic, but also for the ocean. And here you have a very interesting publication on phage interference, the function of the phage T4 immunity and speckle genes and genetic exclusion. So he writes, genetic exclusion is the ability of a primary infecting phage to prevent a secondary infecting phage from contributing its genetic information to the progeny. The two genes in the phage T4 mainly responsible for the genetic exclusion are the immunity IMG and the speckle gene. So here you have the story. This is the Escherichia virus T4, which is a bacteriophage, which interacts with a prokaryote or Escherichia and interacts but in this representation, it's very difficult to see what's happening. So I explain this Escherichia virus T4 bacteriophage within my way of drawing virus-provirus interaction. And so we have virus, which is bacteriophage, attaches to a receptor and triggers what happens in the Escherichia, which is a cell. And mind you, the bacteriophage is a virus, and the whole whatever they say about bacteriophage is a theory of virus and cell. So let's see what happens. We start with the primary infection. So we have the primary phage, which is a virus, as I said, attaches to a receptor and triggers the formation of a provirus. It's called here prophage. And everything is similar to what we have talked before. But the essence of the exciting story is that if a secondary phage attempts to infect our Escherichia, it activates the provirus, but he cannot infect it. And this is the genetic exclusion in the ocean. So again, I remind you what happened in Rubin's lab. You had ALV was the first infection, and the RSV was the second infection in terms of oceans, and you have viral interference in the ocean. Very exciting, and I advise you to look into it. So conclusion, virus is a cell messenger created by the cell, and no cell means no virus. More to come. Enjoy.